Pleasure to introduce Adam Phillippy as our first keynote speaker. So Adam is the head of the Genome Informatics section and senior investigator in the Computational and Statistical Genomics branch at NIH. Adam is a bioinformatician who bridges the fields of computer science and genomics, and his lab has developed many widely used tools for genome assembly, alignment, clustering, forensics, and metagenomics. It is likely that everyone in this room has used his tools, used a product of his tools, or was otherwise inspired by his work. Adam entered the field as an undergraduate when he received his bachelor's in computer science from Loyola University in 2002. There he was advised by Dr. Art Belcher, shown here, along with a much younger me, Adam, and Adam's littlest girl.
And that baby is now 17 years old and going to college next year.
This connection to Art as an undergraduate led him to interning and then later joining the Institute for Genomic Research, Tiger. At Tiger, he worked with Art, Steven Salzberg, who I think is here or online, me and many others, and on some of the first sequence alignment methods and variant calling methods able to compare whole genomes. I think this is where Adam also first cultivated his attention to detail in genomics that has really served him well throughout the rest of his career. In 2005, Steven Salzberg recruited Adam to the graduate program at the University of Maryland. This was a truly awesome experience. Working together in this group was incredible. We pushed each other harder and further than any of us could possibly imagine. It's fair to say that everyone there benefited enormously working in this incubator for innovation. We did have a little bit of fun at times.
I very nearly wore that same shirt tonight, and I'm glad I didn't.
We did manage to get a little bit of fun in at times. Here's Adam flying a drone around our little office. He also was a big juggler at that time as well, if I recall. After graduate school, Adam advanced directly into a PI role at the National Bioforensics Analysis Center. There, he and his group pioneered the use of single molecule sequencing and min-hashing for the comparison of large genomic data sets. For example, here's Adam with Hilary Sussman. She's the editor-in-chief.
Oh, it's the same shirt. You did this intentionally.
We got to spruce up your wardrobe. Editor-in-chief of Genome Research to celebrate a special issue that Adam edited about these methods. This included Canoe, which has since been cited several thousand times for its unparalleled accuracy, speed, and simplicity. In 2015, Adam joined NHGRI, where he has worked hard as a thought leader and as a consortium leader in several major projects, such as the Vertebrate Genomes Project. In my mind, the VGP is the most ambitious and most important project ever considered to understand human evolution. Even more recently, Adam, along with Karen Miga, shown here, co-founded the Telomere-to-Telomere Consortium with the goal of finishing the human reference genome. Initially, I think this was foreseen as like a decade-long effort, or better.
A career-long
effort. But under Adam's leadership, we were able to accelerate the project to finish many years in advance and with quality far beyond what any of us could possibly imagine. For this work, Adam, on behalf of the entire TET Consortium, was named by Time Magazine as one of the most influential people in the world. On a very personal note, I would just like to say working with you in the consortium has been a true highlight of my career. You are incredibly brilliant with your ideas, incredibly generous with your support, and incredibly driven to take us places that we never thought we could reach. So thank you for your friendship, thank you for your leadership. I'm incredibly excited to hear your keynote. The human genome is finally finished. What's next?
Oh man, thank you, buddy. Bar none, the best introduction I've ever had to a talk. Thank you, Mike. Goes back a long way, and a lot of the stuff I'm working on now was inspired by Mike. I remember he was doing genome assembly at the time I was doing genome alignment, and I clearly recall telling him at one point, I'm never doing genome assembly again. It's so annoying. I'm sick of it. It's too hard of a problem. Came back around, and here we are. So thank you, Mike, also for giving me a second chance at this. I gave a talk at Genome Informatics last fall about this project, and it was like a 15-minute talk. I tried to describe the whole human genome in 15 minutes. It wasn't my best work. I have an hour today. Hopefully we'll do a better job of it, and I'll walk you through the tremendous accomplishments that this consortium has achieved over the past few years. So the big question is, why wasn't it finished in 2004 when Bill Clinton, Francis Collins, Craig Venter, everybody kind of announced the Human Genome Project was done? Well, it's a very simple reason that repeats make puzzles hard. So when we're doing a genome, we fragment it into many little pieces, and we have to put it together, again, computationally, just like a one-dimensional jigsaw puzzle. And when your puzzle's filled with Waldos, that makes it very hard, especially if your pieces are small, because every piece you pick out of the box looks like Waldo. And you'll probably start a puzzle like this on the giant and the dragon and put those together and maybe give up because the rest is too hard. I think it was Jason Chin that tweeted this puzzle at some point a few years ago, saying that he thought it was apropos for this because the Waldos have these stripy shirts, which kind of reminds us assemblers of tandem repeats of red, white, red, white, red, white, red, white, and those are like the worst thing to assemble, and so Waldo gets the cake for the worst assembler. But the real question and why we started this T2T project is, what are we missing? Yeah, we know the Waldos are there and there's a lot of them, but there's stuff buried in this puzzle. Like, for instance, there's a wizard somewhere in that puzzle. Can you spot the wizard? It'll take you a while. He's buried down here at the bottom. And so if you think of these wizards as like, the functionally relevant or disease association variants within the genome, if they're buried within all of these repeats and we haven't assembled them well or they're not included in the reference, we're going to miss them. All of our GWAS studies, all of our variants aren't going to be called against them, and we won't be able to make those associations or understand what's going on. So we were tremendously grateful to have finished this project and published it this past April, announcing that the human genome was finally done from telomere to telomere, all of the chromosomes. And it was really thanks to the tremendous technology development that took place over the past 20, 30, 40 years in the sequencing field, that the Human Genome Project really spanned from 1990 roughly to 2000, a 10-year time span to get the first 92% of the genome done. But we've been waiting around for 2 decades to get that last 8%. And the reason we had to wait is because our puzzle pieces remained, actually the puzzle pieces got smaller for about a decade before they then got bigger again. And now with this great technology in hand, we're finally able to do it. And we were able to solve the genome with this combination of both HiFi and ONT ultra-long sequencing. And this is actually an infographic on the cover of Science. Each of those red bits inside of the chromosomes are the regions of the genome that this project finished, and it totals about 200 megabases, or 8% of the genome. And really, we've been fooling ourselves for the past 20 years. Anytime we talk about doing a whole genome analysis, we've been doing a 92% genome analysis, and we want to know what's in that 8%. So here it is again in a linear form, and it was really Sergey Nurk, who's now at Oxford Nanopore, but was a postdoc in my lab at the time at NIH, that really drove this project over the past 3 years and took us to where we are. And what's missing is all here. That's misspelled. Repeats is what's missing. Hopefully the genome is more accurate than my slides. Centromeres, telomeres, segmental duplications, tandem gene arrays, the short arms of the acrocentrics, which we'll talk a bit about. And as I said, 200 megabases. In there is about 2,000 new gene predictions. So the question is, do we really need that 8%? We were told for the past 20 years the human genome was finished, and we've done tremendous work with the 92% that we had. Well, the short answer is without that 8%, you're dead, right? This is the centromeres. They're pretty important. This is the rRNA genes. They're pretty important for the ribosomes. You need this sequence. And I like Sergey Korin, who's here, a longtime associate investigator with me at NIH. This is his Korin test of assembly quality, that if I synthesize my assembled genome and make new synthesized life, do I get the same organism back that I assembled? And so imagine you sequenced the woolly mammoth and you synthesize that genome. Do you get the same woolly mammoth back? And maybe not if you're missing that 8%. So bottom line, repeats absolutely matter, and they have been really given a bad rap over the past 20 years. Not because they're not important, but because we've really had no way to assay them, to look at them and understand what they're doing. And so I think that's going to change. That's the next decade of genomics is going to be repeats matter. And so I'll talk about a number of these, but just to kind of get everybody on the same page, when we talk about segmental duplications, this is a sequence about a kilobase or larger, 90% identity or larger that's copy-pasted from one region of the genome to another. We have gene arrays, so these are genes that are tandemly duplicated, usually head to tail in a fashion like that. The rRNA genes and the so-called rDNA units are a good example of this. You have haplotypes. Most people don't think of these as repeats, but it's really a giant 2-copy repeat in your genome that's very, very identical to one another, and that makes it difficult to assemble, and it's why phasing is a challenge. You have the centromere higher order repeat structures, which are actually tandems within tandems, which is like Waldos made of Waldos, and it's incredibly difficult to assemble these. And I personally had no hope we were going to be able to assemble these centromeric repeats even 5 years ago before this project really took off. The telomeres, canonical T, T, A, G, 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 repeat. And then transposable elements, and I didn't really know what kind of graphic to make for those, so I made a meme, TEs. TEs everywhere. The genome is just like solid TEs, like 50% of your genome is TEs. It's ridiculous. And so repeats matter. So I'll go through a couple of examples here. The first one is chromosome Y, which was the last human chromosome for us to successfully assemble because it's the most repetitive of the human chromosomes. And thanks to Sergey Nurk, who spent a summer working on assembling this, and my staff scientist, Arang Ri, who's with us all here today. If you have any chromosome Y questions, please find Arang and ask her. More than 50% of the GRCH38Y is unknown, just N characters for more than 50% of that. The majority of that is this giant 35 megabase pair, as we uncovered, human satellite 1 and human satellite 3 mosaic array, shown here in yellow and blue, where yellow is HSAT3 and blue is HSAT1. And we've since looked at many more human genomes, and they have this same really cool mosaic pattern. And the sizes of these individual units changes and the number of them change, but they're always roughly in a 50-50 ratio, which is kind of interesting why they balance like that. It might have something to do with the overall density of this region of the chromosome. This is showing all the alignment differences between 38 and our new complete Y. Some of these are real biological inversions and duplications, but also when you see these big insertions here, this is stuff that was missing from the reference that we've now completed and assembled, and you can see that it's very gene-rich. I'll talk about that on the next slide. And if you look down here, it's a bunch of the repeats that we're interested in, and you can see the pseudo-autosomal region at the beginning of the chromosome is enriched for sign elements, and then non-sign TEs in everything but the pseudo-autosomal region, and then this giant HSAT array. But what are these genes that I'm highlighting here? Well, one of those is TSPY. This is for testes-specific protein Y. And it's a gene that has really undergone an expansion relative to other primates. CHM13 chromosome Y here has a TSP array with 45 complete protein coding genes, shown here as the little red ticks. GRCH38Y has 6-ish complete copies, a couple of pseudogenes, and a giant gap in the middle. And it makes it very hard when you're mapping other TSPY reads to this array because you've got a gap and misannotations. When you look at an actual complete human genome, it's very homogenized, complete array of all complete functional genes within there. But when you really zoom into this, this is one of these tandem gene arrays. All of these little colored boxes are transposable elements or other repeats, and the TSPY genes are buried within this big array. So you have all these transposable Waldos walking around, and you've got your wizard sticking right in there, duplicated many, many times, and doing something that's very important, presumably for spermatogenesis. We have a tool that Alex Sweeten is presenting in a flash talk after this, so I won't talk too much about it other than to say it uses a hierarchical motomizer concept to give these very nice so-called stained glass tandem repeat plots. This is the chromosome Y centromere right here. And so picture this like an alignment dot plot, but the colors are the identity or the conservation. So dark red triangles mean very conserved and homogenized tandem repeats, and the blues are more diverged arrays. This is a very useful tool for analyzing some of these very repetitive regions of the genome. Importantly, because this is alignment-free and based on motomizers, you can generate these plots now for entire chromosomes in seconds, whereas it used to take hours in some cases to do that alignment with Minimap2. A slide that I've included specifically for our advisor, Steve Salzberg, who cares a lot about contamination. Having a complete human genome is revealing that microbial RefSeq has a lot of human contamination in bacterial genomes. And so the typical process in the lab is you culture up your bacterium and you sequence it, and while you're culturing it, you're shedding skin and spitting into your culture plates and filling it up with human DNA. You sequence your bacteria and you assemble it, and then you say, okay, I have contamination in my genome. I'm going to match it against GRCH38, throw out all the human reads, human contigs, and I'll have a clean microbial genome. Well, your human assembly was missing 8%, so you only screen 92% of the human genome. And when you look at, on chromosome Y specifically, these are 64 mers that are only in our CHM13 complete Y and that show up in RefSeq microbial genomes. And so this is the number of RefSeq microbial genomes it shows up in. This is the position on the Y chromosome, and you can see a ton of microbial genomes have these human satellite repeats in their assembly. This is the length of the contigs. They're all typically, as you would expect, really, really short contigs, and this is all of the different genre that it's spread across in microbial RefSeq. And RefSeq is supposed to be our best database of microbial genomes, but you have Vibrio, Bacillus, Escherichia, Acinetobacter. All of them have human satellite DNA within them. That's not biological. That's purely lab contamination. And now that we have a huge, complete human reference genome, we can uncover all of that, clean up the databases. It actually can result in some really insidious consequences. There's this cool paper from Chrisman et al., I believe at Stanford, on the human contaminome. And they were looking at human sequencing studies, and they saw a differential abundance of microbial genomes called in men versus women. And we're wondering, what's this differential abundance in these calls? And it all comes down to the fact that men have Ys, and all of those Ys were falsely hitting microbes that were contaminated in RefSeq. So beware of microbial contamination and human contamination as well. Next up, the segmental duplications. So I use an example here from king of segmental duplications, Mitchell Volger, who was at the Evan Eichler lab and studied this in the context of the T2T project. And in his companion paper that came out in that Science issue, he highlights this one particular gene, TBC1D3, which is known to be important in cortical folding and creating outer radial glial cells. Not just CHM13 at the top here, but Mitchell and colleagues assembled that same gene array in 17 additional human haplotypes and uncovered large copy number variation within them. So segmental duplication, copy number differences go hand in hand in this case. And so the way you interpret this plot is that the CHM13 reference is kind of this solid increasing triangle. And as that gets rearranged, new sequences go in here with different colors. And if there's an inversion, the triangles kind of switch their slope the other way around. And these are all human haplotypes, haplotype resolved as de novo assemblies. And you can see that the number and orientation of these genes is all over the place. And so it's known that this region of the genome has undergone a big copy number expansion relative to chimps. And so it's thought that it has something to do with the increase of brain size in humans. But when you look at humans, it's also all over the place, and all of our brains are the same size. So what's going on here? It's kind of at odds with the previous description of this gene and will require future studies. But, you know, to Shell and others that are working on autism, a lot of the neuronally development-associated genes have this feature of being high copy number and copy number variable, and having them completely assembled in a way that we can analyze them is hugely important. And so when you look at CHM13 and you look across the genome at all of these copy number variable genes, it turns out that CHM13 is a way better reference for understanding copy number variation than GRCH38. Mitchell and colleagues went back in and copy number genotyped 268 diverse individuals from the Simons Genome Diversity Project and found that CHM13 is 10 times more likely to represent the true copy number of any of these individuals than GRCH38, as plotted here. So pick anyone out of this room, and CHM13 is 10 times more likely to match you than 38. And that's because 38 has a large number of artifacts within it. And as I said, that holds for everyone across the world. This isn't like an ancestry-specific effect. The red bits of these pies for the SGDP show where CHM13 is a better reference, and you can see for all of the populations in that cohort, CHM13 is a better representation of copy number in the genome. Not just a better representation, but a whole lot more sequence. 60% of the segmental duplications in CHM13 are either new or structurally different from HG38. That totals 25,000 new SDs, or 80 megabases of new segmentally duplicated sequence. And that's shown here in red. In this case, this is only showing intra-chromosomal SDs. There's also interchromosomal SDs we'll talk about in a bit. But the ones I want to talk about in particular are these ones on the short arms of the acrocentric chromosomes. And you can see red is new SDs, and those regions are very, very enriched for new SDs in this assembly compared to HG38. And there's a very easy answer for why that's the case. There's no sequence in HG38 for the short arms of the acrocentric chromosomes. And so, of course, everything that we've found there is new. And so repeats matter, the acrocentrics. Why do the acrocentrics matter? Here's a nice fish plot of the 5 acrocentrics in CHM13. This was by Jen Gerten's lab, specifically Tamara Potapova. And if the colors on this look similar to your abstract book, it's because the cover of your abstract book is CHM13 as generated as a fish image from Tamara Potapova. In this particular instance, the rDNA arrays are highlighted in pink. And this is really the easiest thing to point to as the most important function, so to speak, of the acrocentrics, is that all of your rDNA units functional in your genome exist on the short arms of the 5 acrocentric chromosomes. So hugely important for the functioning of a genome, and they're also involved with the formation and maintenance of the nucleolus in the cell. The rDNA units themselves are copy number variable, as you can see from the colors on this plot, but to date, this is about the best resolution view we had of those short arms. We didn't have any sequence, minus a couple of back libraries here and there around the rDNAs. No idea of what the actual structure of the seg dupes and the satellites look like within. So the next slide is going to show it from the telomeric end of the short arm through the rDNA array and up to the centromere, and we're not going to talk about the long arm because that's euchromatic and looks like the rest of the autosomes. Here's what's there in the short arms of the acrocentrics. This is the telomere on the left, the centromere on the right, and the coloring is showing you the different types of satellite repeats that are in there, and gray is the segmentally duplicated sequences. And what I want you to take away from this figure is that it's a mosaic of satellite tandem repeats of all various classes sandwiched in between segmentally duplicated regions. And those segmentally duplicated regions are very gene-rich. In pink and green are the gene predictions across all 5 of those acrocentrics. And anywhere the seg dupes go, the satellites seem to go too. We always see them hand in hand throughout the genome, and so there's some relationship there that'll be fun to understand better from an evolutionary perspective. The one thing that I find most striking about the acrocentrics is that they're very unique in that anywhere you look in the genome, you can really tell what chromosome you're on just by looking at the sequence. But the acros are different. If you look at the rDNA gene itself is nearly identical no matter which chromosome you're on. And there's other regions like this large segmentally duplicated region on 13, 14, and 21 that's well over 99% identical between the 5 chromosomes. And that's the only place in the genome that has that level of similarity between different chromosomes. And as we're looking now at not just a single genome, but starting to look at more and more human genomes, we're finding that a region that might look like chromosome 13 on CHM13 actually looks like chromosome 14 on some other individual. And so we're starting now to see evidence for active recombination between the short arms of the acrocentrics in the living extant human population, which is really interesting when we think about how we're representing human reference genomes going forward. If my chromosome 13 looks like your chromosome 14, a linear reference doesn't cut it in those cases. So to summarize what we found on the acrocentrics, it's about 66 megabases of new sequence, 1,500 of the genes. So really, the vast majority of the new genes we found in this project are located on the acrocentrics. Most striking, pick any 5 kilobase window out of any of those acrocentric chromosomes, and none of them will be unique at an 80% identity threshold. That's how duplicated the sequences are on these chromosomes. And not just that there's no unique one, the median identity of any of those windows to some other window is almost 99%. So kind of highlighting that there is recombination happening here. It's the only way you would have this level of conservation across them. Not just within the acrocentrics, but if you take any of those 5 kb windows, you can find 96% of them somewhere else in the genome, not on the acrocentrics. And that's shown by this Circos plot with the 5 acros here and showing that that sequence is distributed all across the genome. So these are very dynamic sources and or sinks of material, segmental duplications and variations across the human genome, and it's going to be super, super cool to look at the population variation of these regions within humans, and then from humans to primates and other species, and understand how this all came about and what its possible functional role is. So the T2T genome, the human genome, is finally complete. This is the most bases of a single incremental version of the reference assembly ever. And by all metrics, CHM13 is a better, more representative reference than HG38. And the new sequence that we've uncovered is already enabling new science. But we were very careful in the title of this paper. It's the complete sequence of a human genome. This is a single haplotype of the human genome. And obviously, we have 7 billion human genomes on Earth. And so it's just a start. We're headed really towards a multi-reference future. There's no way a single haplotype can represent all of us. And we'll talk about that next. But for the near term, we have CHM13 plus a Y chromosome available on UCSC Genome Browser and NCBI. It's called HS1, and it's available as a top-level genome on the browser. So what's next? And note to self, for me, I got to breathe. All right. This is about the halfway mark. I think I'll be able to get in the rest of this talk. My clock says I still have about 20 minutes left, so we'll do it. So what's next? The reason I titled this talk What's Next is because ever since we released the human genome and we published it and it's finished, everybody always asks me, that's their first question, so what are you going to do next? even my own mom and dad, like I go home for the holidays, they're like, well, what are you going to do now? You've been in the time 100. You know, it's a really, really annoying question. Because as Mike said, we thought this would take us like 10, 20, 30 years. I could just like, you know, my whole career would be built on finishing the human genome. And then 4 years later, we were done. And it's like, oh, crap. What am I going to tell NHGRI I'm doing now? Well, the very near-term solution is we have to automate this. So this process still took on the order of about 3 or 4 years of sequencing, methods development, assembly. And we cheated. The CHM13 genome that's on your booklet is diploid, but it's almost completely homozygous. There's only like a few thousand heterozygous variants in that genome. So we really have to extend our methods to do diploid assembly. And that's where this fantastic team of developers with me, Mikko Rautiainen, Sergei Nurk, Sergei Korin, and Brian Wallens come in, develop this new pipeline called Virco, which takes all of the knowledge that we learned in doing this first complete genome and put it into an automated package. And the way we're doing this is the same recipe that we used for CHM13 of combining PacBio hi-fi reads with ONT ultra-long reads greater than 100 kb, and we go relatively deep for all of those, so 50x coverage. And by that I mean 50x coverage of reads greater than 100 kb for the ultra-long. So currently that's maybe about 5 PromethION flow cells, totally doable for a genome you care about. And then we get Illumina data from either the parents if we have a trio, or Hi-C data of the child if we don't have the parents. And we build a sparse multiplex de Bruijn graph building off of work from Anton Bankovic in his LGA assembler. And then we use a read to graph alignment approach to take all of that juicy long ONT data, map it against our graph, and resolve the remaining tangles. And at the end of this process, we get a bunch of diploid phased T-to-T chromosomes out the other end. If you want to know anything about Virco, Sergei Korin is with us here at this meeting, and he has a poster later this evening. You can ask him any questions you have about Virco. But I'll give you a little overview of why this works. And so this is an actual Virco graph of human chromosome 1 from the Genome in a Bottle HG002 genome. And each of these little noodles in this bandage view is a piece of DNA, piece of sequence. And when they have these so-called bubble structures, that's the technical term for these, that's one haplotype from your mom, one haplotype from your dad, and they have enough differences between them that we can tell them apart. And the whole mic just fell off. Getting too excited here. It's okay, it just doesn't hang on very tightly. Okay. And the bits in between the bubbles are the homozygous regions where mom and dad look very similar because they have some shared ancestry. The ONT data is very, very handy for making these graphs as phased as they are. As an example, these two haplotypes are phased out nicely in the Virco graph after you've integrated both the hi-fi and the ONT. But if you look at this region, kind of near the centromeric region, in the hi-fi only data, it's a lot more bubbly. The phasing is less complete. You have a lot of homozygous nodes. There's a lot more noise. There's repeat tangles. Once you integrate the ONT data and align it to this hi-fi graph, you can resolve all of that complexity and get very nice, clean, assembled haplotypes. That has the added benefit that those really big bubbles are easy to phase across the whole chromosome. So if we come in with Kamer marker data from the mom, the dad, or Hi-C data or strand-seq data, we can map those haplotype-specific Kamers to this graph, and these are like multi-megabase scale haplotypes that are already phased. It's very easy to find those markers in those and then color this whole graph by haplotype, where we have mom in red and dad in blue. Once you have these diploid assembly graphs colored by haplotype, then it's just a matter of walking the paternal haplotype, in this case, from telomere to telomere. In places where it's homozygous, you just duplicate that sequence into both of the outputs, and you get complete T-to-T haplotigs for the paternal haplotype and the maternal haplotype for most of the chromosomes. And so our big announcement is that we've done this now for HG002, and we have a complete diploid assembly for all of the chromosomes of this important Genome in a Bottle benchmark line. And this is the topic of Sergei's poster number 103 later tonight. And this is what the whole Virco graph looks like. This is before you actually phase all of these chromosomes out, but after we've assembled it and labeled them by haplotype, and you can just see the blue walk is just very clear through that graph, and you walk end to end, you get complete assembled haplotigs out the other end, with the exception of the rDNAs, which are forever my nemesis, but we'll get to those eventually. So with these methods in hand, where we can now get automated T-to-T assemblies, we're looking forward to building a human pangenome. So what's a pangenome? Well, the consortium that's come together is funded by my institute, NHGRI, to sequence 350 ultimately T-to-T assemblies using this recipe of hi-fi, O and T, trio data or Hi-C data, combining those into what we call a human reference pangenome, and then building new reference data structures and ecosystems of tools around those. But what is a pangenome and why do we need it? What does it mean? Well, my biggest pet peeve in this space is that there's no such thing as a graph genome. And so if any of you talk about graph genomes on Twitter, I will hound you and I will tell you not to call it a graph genome because it's really just a collection of haplotypes and or variants that better represent a population. In the end of the day, all of your genomes are linear. And that's what we're trying to collect here, these linear haplotypes from all of your chromosomes. And the reason that we want to do this is because it will alleviate bias by replacing a single genome that can't possibly represent everyone by a collection of genomes that's more representative of the genetic diversity in the human population. And once you have that, I'll show how this yields immediate and really free improvements in variant calling accuracy and comprehensiveness. And so if you take one thing away from the pangenome, in this framework, it's really just a set of haplotypes. The reason the graph creeps in is because once you have that set of haplotypes, it's very, very tempting to align them all to one another and find all of the homologies, and the graph is the representation of that alignment. So when you talk about a pangenome graph, that's a pangenome alignment. The pangenome itself is this collection of haplotypes and variants. So on the topic of alignment, which is very near and dear to my heart, Bryce has a poster 102, I'm not sure if that's tonight or tomorrow, showing how we've readapted our MashMap tool to alleviate some of the bias that was in the minimizer-based approach, and we now use a sliding min-hash-based approach. This both made the method faster and more accurate, and we're working with Eric Garrison to use MashMap as a pre-aligner for his WF-Mash and Pangenome Graph Builder tool that we're using to paste together these alignments and build nice pangenome graphs from all of these human genomes. So check out Bryce's poster. We have a preprint up as of this summer on a draft human pangenome reference. This is just in the near term, 48 initial samples as we work up to the 200 plus ultimately. And the samples were chosen from the 1,000 Genomes Project collection, which are immortalized B cell lines that you can just order from Corel, and a lot of trios available from diverse sets of populations. In the first round, for the first 48, we specifically targeted underrepresented populations in current genomic data, and we checked that they had good karyotype, they were low passage, so they don't have a lot of somatic variants building up in them. They have trios and parental data, so we're up to about 100 assembled human haplotypes and counting. Now, these aren't all complete T-to-T finished genomes, but they're really darn close, and each one of these, I would argue, is better than the current HG38 reference in terms of both accuracy and completeness. And so once you have this completed pangenome set, and you look at some of the copy number variable regions, just as an example that we used in the paper of the Rh blood group antigens, this is the count in the data sets and the allele frequencies corresponding to that, and then here's kind of what you would be used to as a linear plot of the genes, showing all of the different structures that are present here and how those are represented in this alignment represented as a graph here. And so the idea is that if you come in from one of these more rarer haplotypes and you're trying to genotype yourself against a reference genome, no longer are you forced to kind of match against the most high frequency variant, but there'll be something in the collection that probably matches your haplotype as well. That'll improve all of the accuracy of the downstream analyses and remove this bias that's inherent from this one reference paradigm that we've been in. And it's not just long reads that benefit from this. Even if you have old short read data, you can use tools like Giraffe to map that short read data to our pangenome collections. When you map to the pangenome with Giraffe and project back to HG38, you end up with 34% fewer false positives and false negatives than kind of current state-of-the-art mapping against HG38 and running deep variant. And so you get kind of best-in-class accuracy of small variant calling, and you get many, many more structural variants called, because then you can start to really impute structural variation from the graph, even with short reads. And so when I say we get free benefits from the pangenome, you don't need to know that under the hood there's a graph. It's in there. You're giving it the data. It's using the graph but not telling you about it. You're still getting variant calls back with regard to whatever reference you want, and those variant calls are more comprehensive and more correct. So it doesn't just stop with human. What's next? All right, it's a very unoriginal research plan, but we're just going to T-to-T all of the things that we possibly can. And the first ones that I'm most excited about are the primates, in particular the non-human apes. And so announcing the T2T Primates Initiative in collaboration with Katarina Makova at Penn State and Evan Eichler at University of Washington, as well as a tremendous group of folks that have contributed these samples. We've replicated the T2T sequencing recipe and generated Virco assemblies for bonobo, chimp, gorilla, 2 species of orangutan, and now 2 species of gibbon. And for bonobo and gorilla, we actually have trios, I think in this case probably thanks to the San Diego Zoo. And so we can take all of these primates and run them through Virco and get near T2T assemblies. And within the next couple of years, we'll be able to make all of these as high quality as CHM13 and ultimately gapless. These draft assemblies are available now. Chromosome X and Y for all of these primates and humans are actually the first ones that we've completed and validated, because that was primarily my interest and Katarina's interest in starting this project. But for the autosomes and for the whole genome, we have an average of 36 T2T haplotypes per these species, and all of the other chromosomes are in just a handful of remaining pieces. And this is the Virco graph for bonobo, and you can see that almost all of these chromosomes are split out as complete haplotypes. There's very few bubbles left, and we're very, very near, straight out of the box of Virco, of having a T2T bonobo genome. So we have a poster here from Brandon Pickett that's primarily focused actually on a panel of 10 complete human X chromosomes that he worked on over the past year. But with the primate project taking off with leaps and bounds, we actually have added all 6 of those non-human primates and have complete T2T X chromosomes for them as well, which will enable really, really cool comparative genomics, not just within the human pangenome, but extending out to the ape chromosome X pangenome. So I think I'm going to make it. The countdown still has 3 minutes, and I'm on the wrapping up slide. We can do it. What now? All right, this is just the summary of what I talked about. So this is kind of what's going on now. We've automated T2T assembly for all but a handful of the most difficult chromosomes. We're building a T2T diploid reference genome for HG002. We're building a human pangenome with these methods, and we're repeating for important model organisms, not just primates, but there's a bunch of associated T2T projects kicking off for important model organisms like zebrafish, mouse, fly, agriculturally important genomes like the ruminants and so forth. So with these in hand, like, why go to all of this work? Well, we can finally read the complete code of life. I always like to think of the genome as, the code, computer code of life that gives you the instructions to build the genome, live a happy life, and so on. What do we do now that we can read that complete code? Well, the one thing that when I say most people realize, I'm really talking about myself here, that once we finish these genomes, I am seeing way more structural variation, gene conversion, recombination between chromosomes than I ever pictured in my mental model of how the human genome works. And I think that that's going to surprise a lot of people once we have these T2T genomes, how frequent, recurrent, large-scale multi-megabase inversions are in the human genome, recombination across chromosomes, and large structural variation. The old trope that 99.9% similar is humans. At the SNP level, when we look at the structural level, I think we're going to find a lot more variation. This is leading me to say that really any quote-unquote whole genome analysis going forward needs to be long-read, because that 8% that we've unlocked here is really only mappable with long-read sequencing. I think only like 1% of that is mappable with Illumina data. You need the long-read data to map it. And when I say whole genome analyses, I mean like anything that's star-seq, RNA-seq, high-C, chip-seq, any seq. Long reads so you can actually map to all of these paralogs and all of this new sequence of the genome. Ultimately, we need to push all of the variant benchmarks to be fully comprehensive, not just looking at the easy to call 90% of the genome, but making them long read based and pushing them to the full 100% of the genome, because I know that buried within that sequence, there are new disease associations and their new biology, and this is what has me most excited about these T2T projects, is that when I first start seeing some new disease associations coming out from this sequence, it'll be tremendously gratifying. So the last thing that I'm going to pitch is just really on this topic of building better benchmarks in this HG002 reference sample. We've now completed chromosome X and Y. The chromosome Y from this sample is actually the one that you'll find if you look at the CHM13 genome browser. These are both complete and validated at Q70, which is 2 orders of magnitude higher quality than the original human finished genome. And we have now draft diploid autosomes for all of these that hopefully we'll have validated by the end of the year. And so we're teaming up with the Genome in a Bottle folks for a project that I call the Q100 project. because these Phred scores are logarithmic. So Q100 means, on average, 1 error every 10 billion bases. And so 6 billion bases in a human genome, Q100 means roughly a perfect human genome. And so I would like to make HG002 the first, quote unquote, perfectly accurate genome, because I think that'll make a tremendous benchmark and really push all of us to make our methods work on the complete diploid genome that all of us have in ourselves. And so with that comprehensive diploid assembly, we'll be able to make assembly benchmarks that can improve genome sequencing technologies and improve variant calling, really with this goal of not reporting 99% accuracy on a genotyping assay when you're only measuring 92% of the genome. Any variant calling that happens right now should have like an automatic minus 8% because it's not measuring the whole genome. We'd like to push that all the way. So I'll end with a slide on the resources. You can find a lot of this on my lab's GitHub page, which is Marble. The Virco assembler is there. All of the CHM13 data is there. All of the HG002 data is there. The Human Pangenome Reference Consortium you can follow on this website. And then all of the data for the primates and all of these other model organisms will be hosted at the genomark.github.io, which is affiliated with the Vertebrate Genomes project that Mike mentioned earlier. So I'll stop there, but not without first acknowledging Karen Miga, who really had this idea with me back in around 2016, 2017. Let's just finish the genome, and we were naive enough at the time to think we could actually do it, and so we undertook that project, got CHM13, sequenced it to death, and with the help of these wonderful people here in my lab, actually made it happen. So the folks in green are here at this conference. I encourage you to check out their posters. And a number of folks from the wider T2T consortium are here as well. This is us celebrating our first in-person meetup back in August in Santa Cruz after 2.5 years working in our basements during the pandemic. So contact me if you're interested in joining these efforts, and I'm happy to take your questions.
Oh, so that was an amazing presentation. So I'm going to take privilege. I have a million questions, but I'll hold myself to 2.
Yeah.
So and I think I'm speaking for many people in the audience that have been using GRCH38 for maybe years. You told us CHM13 is amazing, but then you just told us HG02 is going to be Q100. Then you told us the pangenome. Like, what should we do? Should we get started? Should we wait? You tell me.
Yes. The answer is yes. So there's one reason that I like the HG002 exercise, that if we want to do a variant calling benchmark, you have to take those haplotypes and align it against something that's also complete. And so we'll have 2 HG002 haplotypes that'll be perfectly accurate. We can map them against CHM13, catalog all the variants, and that'll give us a nice variant catalog against a single linear reference. We can repeat the same thing against the pangenome, and we'll have a set of variants called against the pangenome. So you can't just get by with one complete reference. And so this is really just the beginning of making all of the future genomes T2T and accurate. But that doesn't really answer, like, what do we use as the reference coordinate system when we talk about variants? And I think that's definitely a very open question going forward. You know, are we going to refer to things in the pangenome space as it's positioned this on this haplotype? Are we going to project everything to CHM13 and report it as this to this? For the pangenome paper itself, we actually projected things onto HG38 as to not rock the boat too much. And HG37 is even possible as well for the clinical folks that still like to use that. And the reason that that still happens is because there's a tremendous amount of metadata that's on those fixed coordinate systems. And so if you want to look at nomad variants, you have to look at them relative to the genomes that they've already been called on. We can put some Band-Aids on things in the near term and lift over all of those positions to CHM13. The lift over always doesn't work, and you only lift over to the regions that already exist in HG38. So to really take advantage of these comprehensive references, that's why we need the long read, functional studies, variant studies. We really have to go back and redo all of those variant studies and redo data sets like Nomad with long read, functional data, variant data in the future, and build those up on these complete representations. So I'm not, naive here. It's going to take another decade of work to put everything on top of these complete pangenomes. We're laying the foundation of the data here.
Awesome. And my second question, I also think I'm speaking for everyone. Do you stay in touch with Zendaya?
I did not personally meet Zendaya. I did not have the nerve to walk up to Zendaya, but my wife did walk up to Zendaya along with Mike's wife, and I don't know if they still text or not. I'm not privileged to that.
All right, let's open it up for the audience. There's one in the back here.
Wonderful. Thank you for the contribution to the field. This is amazing. I have a reverse question to this leftover thing. So how inaccurate is it to take positions from HG19 or regions rather in HG19 or 38 and try to go to T2T? What would be missing?
So a large fraction of it is liftable in that direction. Obviously, you can't take stuff as easily on CHM13 and lift it back to things that are missing, 8% of the sequence. But a lot of the variants or positions on prior references are liftable to CHM13. The only place that you run into trouble are on these copy number variable genes and within the segmental duplications where there's not a clear one-to-one orthology relationship from the old reference to the new reference because they represent different haplotypes at those positions. But there are some real bugaboos in the lifting process that people don't always think about. And it's in cases where there are artifacts and errors in the old references, and people have mapped data to those old references and called variants within an artifactual region of the reference. And they've associated diseases with those artifactual variants because they are, within the same haplotype and they're in linkage. But then when you have an artificial variant, how do you lift it to a correct reference? And so there's these little corner cases like that aren't easily solved. A clinical database has variants with known disease associations, and we can't lift it because it's associated with an artifact. It's tricky.
Thank you.
Other questions?
You may have implicitly addressed this, but this is something I was curious about. So I know that there are issues, I guess I would think that there are potentially issues with the detection of things like circulating DNA that's like very low prevalence or a low prevalence somatic variants like near the traditional NGS, their prevalence being near the error rate of traditional NGS methods. I was just trying to think if you have anything in mind as to situations like these where you're working with these sort of very small, almost undetectable like molecules in things like cancer and disease-specific things that you want to do in a gene-agnostic way, just like try to do a T2T style investigation on it. Is that like something you're thinking about? Is it something you're worried about? Is it something you can address already?
Yeah, so anything that has small amounts of DNA, you're kind of stuck at the start without being able to de novo assemble a T2T reference because you can't get these huge molecules at the type of coverage that you need to do a true de novo assembly. But the one thing that I think will be very helpful going forward is to do a personalized diploid genome for whatever patient you're dealing with. And so then when you're looking at the short read data or whatever low frequency data you get from that individual, you don't have any confounding variants from the reference being different from them. Any variant you see is an actual variant versus their germline genome. Even with short read data, if you have the exact diploid reference of that individual, you can map a fair amount of sequence that would be otherwise unmappable because all you need is a single kind of defining SNP between the haplotypes. And so I think doing that for any kind of cancer or somatic variation studies, do high coverage of the germline of that individual, get a nice de novo T2T assembly for them, and then map all of the functional data, all of the short read somatic data back onto that, and I think you'll get a much better answer.
There's one over here.
Hi. Assuming you have accomplished the pangenome process as well, do you expect to see sort of patterns of, say, allele segments sort of sorting together, or do you expect a mosaic in most regions for if you were to find a pangenome of some, say, me across the pangenome profile?
What do you mean by a mosaic? So a mosaic of the type of variants?
Right, yeah, of the different haplotype segments that you described.
What kind of segments?
The haplotype maps?
Yeah.
Yeah.
Well, I think it's pretty fairly well understood already in the euchromatin how linkage works and haplotypes are inherited. In some of these repetitive regions, I think the signal is going to be a lot messier because you don't have kind of the same, vertical inheritance of non-repetitive euchromatic sequence that you have. You have gene conversion happening within and between chromosomes and these types of recombinational patterns. The centromeres themselves don't typically have any meiotic recombination breakpoints happening within them. They're inherited as a whole chunk, and it's still to be determined what happens on the acrocentrics, but I presume something similar happens there as well. So the patterns you see on the acrocentrics and in and around the centromeres will be different from the rest of the genome, but I don't think there's any big surprises waiting there.
Thanks so much for the talk. You were talking about the position of a variant in a pangenome becomes, you pick a haplotype and you talk about its position in that or other haplotypes. So having a single coordinate system has been like a convenient fiction. We can represent something in like a bed file, and we can do functional genomics. And so do you have thoughts about, so that explodes, and then we have, multiple coordinate systems for each haplotype in our cohort, say. How, what becomes of the bed file?
Yeah. Well, it's one of the reasons I liked my earlier answer of having a personalized genome for everyone. So if I'm doing an analysis of expression in my genome, I've got my T-to-T diploid genome, I can map to that, and then everything's a bed file with regard to my two haplotypes. Then the question is, how do you relate it back to all the prior knowledge of those orthologous genes in the population? And that's the trickier part. I think if you look at some of the regions of the genome where we already have this problem, like HLA genes, people take a very allele-centric approach of like literally naming every single allele that has a single SNP difference between everything else. And in some of the very variable regions, I expect we'll probably see that crop up as well, that things become more allele-centric rather than variant-centric. But then de novo variants become a whole different ball of wax as well. So I like the personalized genomics angle, and the catchphrase I use is like, you're your own best reference genome. So hopefully we can make the technology so good that everybody has a T-to-T diploid reference for themselves that can go on for somatic variation and functional studies.
Yeah, thanks. You talked about sequence quality, which is great. But what about like gene annotations? You mentioned like RNA-seq and so on. So can I use your like CHM reference to align my RNA-seq reads and quantify gene expression or something like this at the moment?
Yeah, absolutely. We have multiple annotations available on it, generated by multiple independent groups. There's a RefSeq annotation. We generated our own annotation based on some IsoSeq data that we had of the CHM13 line, and so forth. So all of that is up in the browser. So if you would take your RNA-seq data and map it to CHM13 and load it into the browser, you would see it in the context of all of those annotations. The novel bits of the genome, the 200 megabases that are new here, all of the annotations that are there are primarily lifted over from paralogs, known paralogs, elsewhere in the genome. And so it's going to be a little lower quality in the newer regions, and I think it'll take another 10 years to get the annotations improved, especially in the short arms of the acrocentrics. As well, CHM13 is a single cell type, and so the IsoSeq expression data that we had for it that went into the annotation is just whatever genes are expressing in that one cell type. And so adding on additional expression data onto that reference to improve the annotation is a wish list for us, and we'll get there. But for now, yeah, it's a totally usable reference for gene expression studies in the browser.
Maybe one or two last questions.
Hi, great talk. So you mentioned, as most of us know, that there is variation across different individuals, and so the goal would be for every person to have their own reference genome. A question that I've had for a little bit is that good enough? Is there variation within different tissues in the same person that are worth modeling? And is there anything important we should know about that?
Yeah, I mean, I think absolutely there is somatic variation within the different tissues. If you think about the fact that these are all B cells, they've already undergone genomic changes to themselves relative to the germline genome, and so even this pangenome isn't representative of even the germline cells of these individuals. We'll see what we see after we do a few hundred human haplotypes. I think kind of the somatic variation that you would see in the individual tissues probably also exists as germline variation elsewhere outside of things like the immune genes. And so as we make that pangenome representative enough, I think that it'll have a pretty good collection of all the variation that you might see. And a lot of the somatic variants, I think, will happen to be recurrent and kind of around the same repeats, the same palindromes, the same inversions, and so they would probably be captured in somebody's germline genome at some point once you make it comprehensive enough. But Definitely you will have de novos at the point that don't exist in the pangenome, but the pangenome is just to make enough of a representation that you map to it with good accuracy and don't affect all kinds of biases. You will still have to call de novo variants against the pangenome. It won't be comprehensive of every variant that's ever existed in the human population, but then the de novo variant calling becomes much easier if there's at least a haplotype in there that's much closer than the prior reference. But it's definitely a hot area of exploration to understand the extent of somatic variation in the different tissues. And the projects that I'm most excited about in that field going forward are the ones that are doing the germline genome of the individual where that somatic variation is being studied, and the somatic variation is being studied with long reads across different tissues.
Okay, I think we have time for one last question.
Awesome talk, Adam. Really inspiring stuff. So now that you've seen these repeats that we didn't have in the previous references, what are some of the ideas for the purpose or function? You know, if I deleted segments of these long repetitive regions, like from the Y chromosome, would it pass the Corin test and recreate the same me?
Yes and no. Definitely you can delete some regions. It's a question of which ones and how much you can delete without passing the Corin test. With the Y chromosome in particular, that large human satellite array on the long arm varies by megabases between individual males. And so we know that they are really hypervariable in terms of the size of those arrays. But in that case, it was interesting that despite the size, we always saw this 50-50 balance of the two different satellite classes. And so almost like it's trying to maintain that ratio. And so if you disturb that ratio of the human satellites on that long arm, then maybe something would not work as well. But this is, I think, what people are very excited to start digging into now, like with the alpha satellite repeats in the centromeres, like what specifically determines where the kinetochore decides to form, and can you change that, and how specific does it need to be? Everybody that's interested in these types of satellite repeats and the RDNAs are just over the moon about the chance to look at this now and start doing functional studies. And you can take cell lines and start to CRISPR stuff and perturb it and see what happens. So I think that'll definitely be an active area of study going forward. The bit that's most exciting to me is that similarity between some of these segmental duplications on the acrocentrics and what it means to have that gene conversion happening across chromosomes. And there's some studies to see if some of the variations within the RDNAs are like chromosome specific. Maybe you have like specialized ribosomes that, you know, chromosome 15 RDNA array has different variants in the ribosome than the other chromosomes. And we can only look at that now by going across more and more humans. And so those studies are just getting started.
Okay, thank you so much, Adam. That was an outstanding presentation.
Thank you all.